Today we're going to look at pedigree analysis, and a pedigree is normally associated with animals. So you ask for the pedigree of a dog that you're buying that's a full-blooded dog, that you can look at its family history and see who its parents were and who they came from, who sired them, and so on. So we look at a pedigree for a human family. We do that though looking at one particular genetic trait so we can see how that trait has moved throughout history in that family. So a pedigree for an autosomal disorder is going to help us decide if there's a condition that's inherited due to an autosomal dominant or an autosomal recessive allele, and we can get that information from looking at a pedigree. So this is what a pedigree looks like, and we use circles to indicate females, and we have squares to indicate males. And you notice we have a key over here, so the ones that are not colored in are unaffected, and the ones that are colored in this case are affected. So if I were going to read this, I would say that this individual is married to this individual, and they had one offspring that was a female, and the female is affected by this genetic disorder. This, of course, we know this can only happen in complete dominance if both parents are heterozygous for that disorder. So we know that these both must be carriers. So for example, if I wanted to say that this had to do with freckles, and I know that freckles are recessive, and I know that mom doesn't have freckles, and I know that dad doesn't have freckles, but I know in order to see freckles, the genotype of this individual must be little f, little f. Well, that tells me for sure that the parents both have to be heterozygous or be a carrier. So if you have a disorder that is passed on and you don't see it in yourself, you're considered to be a carrier. So if the mother were homozygous dominant and the father were heterozygous dominant, and you see that he doesn't have freckles, she doesn't have freckles, but the child does, in this case, this wouldn't even be a possible scenario because mom would always give at least one copy of the dominant gene. So this isn't a possibility. So the parents are carriers because they're unaffected but are capable of having a child with that genetic disorder. This is an autosomal recessive pedigree, a little bit more in depth than the one we looked at. We cover more generations. One of the things I want you to notice is that you'll have them numbered here in the rows are numbered by Roman numerals. And then usually within the row they have numbers as well. So this one doesn't. We're going to refer back to row 1, row 2, row 3, and row 4. So in this case we have a mother that's affected. She's married to this person. They have 2 children. Both of their children are girls. Their girls are married. Married over here. This marriage produced one child who is a female and is heterozygous. This marriage produced 3 individuals, a girl and 2 boys, and one of the boys married this girl, which they're first cousins, so we don't want to encourage that at all, but that's what happened in this situation. And they had 3 children together. If you notice that affected individual up here, the mother, you start to see that show up down here. So this would be their, I guess it'd be her great-grandchildren, actually, where you start to see this again. So this is why we have to be careful about close relatives who reproduce, because they're most likely to have affected children. And you notice in this case, it didn't matter whether they were boys or girls, they had an equal chance of being affected. In the assignment that you're going to do in class tomorrow, you're going to get more practice on how to set up a pedigree and how to read a pedigree and get the information off of it that you'd like. Let's look at some autosomal recessive disorders. One of them is Tay-Sachs disease. This usually occurs among Jewish people in the United States. This results from a lack of an enzyme that's called hexosaminidase A, or HEX-A. The lysosomes build up in the brain cells, and this results in progressive deterioration of psychomotor functions. So why do we expect to see this mainly among Jewish people? It tends to happen in Jewish people because Jewish people will reproduce with more Jewish people because they tend to marry and reproduce within their religious beliefs. So we see that show up in that group of people. Another one is cystic fibrosis. This is the most common lethal genetic disorder among white people in the United States. What happens with this disorder is that the chloride ion fails to pass through a plasma membrane channel in a cell, and this causes really thick mucus in the bronchial tubes and in the pancreatic ducts. This is a picture of malfunctioning lysosomes in Tay-Sachs. We have the defective lysosome right here and a normal lysosome right here, quite a difference. This is what happens when the protein channel malfunction happens in cystic fibrosis. The chloride ions are trapped inside the cell, and the chloride ion channel doesn't allow those to go through, and that causes this to be sticky, mucousy, and build up here. Another autosomal recessive disorder is PKU syndrome or phenylketonuria, and you were checked for this when you were in the hospital, when you were born, and they would take a blood sample from your heel, and they're looking for the enzyme that's needed for metabolism of the amino acid called phenylalanine. And if you have that enzyme, it's not a problem, but if you don't have it, it can be a really big problem. It can cause severe mental retardation and a lot of other health problems if you have this disorder and you aren't put on the proper diet. So if you are found to have an enzyme lacking this enzyme and a low enzyme level for this, you need to put on a low phenylalanine diet, and that'll be for the rest of your life. Now, before this was discovered that this was what was causing this, a totally normal, healthy infant could be born and fed the formula and then the foods that we have that have a lot of phenylalanine in them, and that would just cause them to be severely mentally retarded. So they weren't born that way. It's something that developed due to the syndrome. Another one is sickle cell disease. This is found mostly in African Americans. These are where the red blood cells are actually sickle shaped instead of regular, and we'll look at what that looks like on a slide here in just a little bit. But instead of being round and foldable and easily move throughout the circulatory system, they're sickle shaped and they're not as flexible. And we've been looking at autosomal recessive disorders. Let's take a look at a couple of autosomal dominant disorders. One of the first ones is Marfan syndrome. This is caused by a defect in an elastic connective tissue protein called fibrillin. There's one also called Huntington's disease, and this is a neurological disorder that leads to progressive degeneration. And the onset of this is normally middle-aged, so people who have this disorder don't know that they have it until they've already chosen to have children. There's a genetic test that can be done to detect whether you carry this gene or not, or have this gene, because it's just one copy that makes it visible. It's a dominant disorder, so they check to see if you have that copy of that gene. And it leads to early degeneration mentally and physically, and not being able to control yourself. It's called the dance because people will try to walk normally and they think that they're actually alcoholics or they're heavy drinkers and they're having trouble walking because of the staggering that they do. They kind of dance around, and that's just an early symptom. That happens early on. It could happen anywhere from 30 to 50 years old, and then it leads to degeneration that is unstoppable. Let's take a look at some normal red blood cells and a sickle cell so you can see how big of a difference it is between those two. This is neurons in a normal brain. This would be neurons in someone who has Huntington's disease. And there's a huge difference, of course, with the degeneration that's happened in here. So what we're going to do in class today is we're going to go ahead and work on pedigrees and look and see how some of those play out, and you'll work with your lab partner on those. And we will get into incomplete and codominance in the next lecture.